So, dear students, good evening to all of you. And now today we have 9th February 2022, and it is a lecture on in the course Ag506. And today we are going to deal with rice diseases and their management. This means not. Eases means ease. Means something is not on ease. Something is not able to remain healthy. It is not in ease. So therefore it is disease. So there are variety of diseases in rice, and some are common. Some come year after year, and some come occasionally. And some do not come in one part of the world, and some may come in other part of the world. So they do not have any fixed incidence. And as usual, disease will lead to the reduced growth of the crop, reduced dry matter production, and sometimes your grain size or grain quality is affected. Sometimes they infect the leaves, or sometimes shoot, whole shoot, sometimes roots. So almost all the organs of rice plants get affected by disease. And these diseases are of two types. One is your non-infectious, other is infectious, means caused by certain organism, and some are not caused by living organisms. So organisms may include your fungi, your bacteria, nematodes, or viruses, mycoplasma, and some other organism can cause some disease in rice. And there are some non-infectious diseases. Some are caused by deficiency or excess of nutrients, and some are caused due to some other changes in the environmental conditions, like changes in moisture or changes in temperature, and some other weather parameters can also lead to certain disease kind of symptoms, or they can reduce the growth of the crop. So today we are going to discuss diseases caused by living organism or bioagents, caused by some biological organism. So we start with bacterial leaf blight. Bacterial leaf blight can...
सर
Hello?
सर अभी ज्वाइन हुआ सर अटेंडेंस सर
Okay.
सर शो को दो
Okay, no problem.
ठीक 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 है सर
Continue to attend the class, please. So you can see bacterial leaf blight, as the name suggests, it is caused by bacteria, Xanthomonas oryzae, PV oryzae. And it causes wilting of seedlings and yellowing and drying. So it can come at any growth stage of the rice plant. This is one. And if it is at seedling stage, the yellowing of seedling can happen and then drying. So when seedlings dry, their color will change to straw color. Areas that have weeds and stubbles of infected plants. So if some stubbles are left over in the field, so this disease can come, and it can occur. It can be severe in susceptible varieties with high nitrogen fertilization. So imbalanced fertilization, particularly in favor of nitrogenous fertilizer, can increase the incidence of bacterial leaf blight. So you can see the symptoms. They are kind of yellow stripes, and then they turn brown. And you can see these long stripes on the leaves. They can be on the central part of the leaves. They can be on one margin of the leaf, and they can be on both the margin of the leaves. So it is not quite fixed that these kind of strips or wavy strips will happen only on the margins, but they can come on any part of the leaf blade. Now, as it is bacterial disease, many of you might be knowing that certain oozing occur in this. So you can see lesions, water soaked, yellow-orange stripes on leaf blade. So you can see on the right side, water soaked lesions. Lesions have a wavy margin and progress towards the leaf base. So they start from the top part and goes down to the leaf base. On young lesions, bacterial ooze resembling a milky dewdrop can be observed early in the morning. So this is a typical identity of bacterial diseases, and it can be tested by certain plant pathologists. They can take the material into the lab and they can identify the causal organism. Old lesions turn yellow to grayish white with black dots. So with time, it will turn to grayish white with black dots due to saprophytic fungi. After certain period, saprophytic fungi will come out to eat the substances which have been released by bacteria. So this is interesting. Then this is kind of secondary infection by saprophytic fungi. Now you see dried up bacterial ooze. Once it has come out from the leaves, after some time it will dry up and it will look like this. You can see the symptoms on leaves, starting from topmost leaves. You can see there is a kind of yellowing of leaves, and then they are turning brown. And you can see the severity in the middle picture on the bottom. You can see the crop is totally dried up. So sometimes it can be very, very destructive disease, bacterial leaf blight. Now, what are the control measures or management options? So one is resistant varieties. So we need to find out resistant varieties, and it is not easy. For a student, it is much easier to write the management option as resistant varieties. But there are hardly any resistant varieties for diseases happen. Because you do not want just resistance in the variety. You also require certain other qualities, certain other parameters in a rice variety. So choice is limited for resistant variety, but it is an option to say that resistant varieties should be grown. First thing is that limited availability, limited choice of resistant varieties. This is number one. Number two, the resistance is not permanent. After few years of cultivation, a resistant variety may lose resistance. So that is the problem with the resistant varieties. But as a student, you can write resistant variety is one option. Other is balanced amount of plant nutrients, especially nitrogen. Here, the excess use of nitrogenous fertilizer should be avoided. Excess use of nitrogenous fertilizer encourages the growth of the fungus and more incidence of the disease. Ensure good drainage of fields. Keep fields clean. As you have seen that old stubbles and some weeds and some other wild kind of plants should be cleaned because they may harbor these kind of organism or bacteria, and then it can come to the crop. So cleaning of the field is necessary. If you have grasses, some wild plants on the bunds, so they should be cleaned. Allow fallow fields to dry in order to suppress disease agents in the soil and plant residues. So after harvest of the crop, the residues should be preferably allow fallow fields to dry in order to suppress disease agents. So fallow field means they must be having some water, and the field should be dried so that fungal propagules may be killed. And at last, if you want chemical control, it is available. 75 gram agromycin 100, this is the name, agromycin 100 is the name of antibiotic, and plus 500 gram Blitox. Blitox is copper oxychloride. Blitox 50 is the name. And these two can be mixed in 500 liter of water and can be sprayed. This spray can be repeated after 8 to 10 days of the first spray, depending upon the severity of the disease. If it is severe, then 2 to 3 sprays of this mixture are required. Now, another bacterial disease is bacterial leaf streak. So it forms the streaks, parallel streaks on the leaf. You can see on the right side. So browning and drying of leaves, reduce grain weight due to loss of photosynthetic area. You can see here, once the photosynthetic area is reduced, the production of dry matter will be reduced, and that will lead to reduced grain weight, transmitted through seeds and infected stubbles. So it can come from soil or field, or it can come from seed. Maximum tilling to panicle initiation is the stage when you will have more chances of its attack. Bacterial leaf streak comes mostly during maximum tilling to panicle initiation. Symptoms are small water-soaked lesions between veins, and streaks are initially dark green and later turn light brown to yellowish gray. Lesions are translucent. Entire leaves may become brown and die. So in the beginning, the lesser area of leaf is affected, but the streaks can increase in their size, in length as well as in breadth. So therefore, entire leaf can turn brown, and in the end, it will dry and die. Humid conditions, droplets of bacterial ooze may be observed on the leaves. Now you can see some more symptoms: linear lesions in the first picture, and then enlarged lesions you can see, and then water-soaked lesions. So there is a very small difference in bacterial leaf blight, commonly called as BLB. So BLB and BLS, bacterial leaf streak. So here you can see that there are the streaks or lines. In between lines, you will find some chlorophyll. So it is not continuous, but in case of bacterial leaf blight, the yellowing part was in one mass, one particular area continuously. But here you can see it is kind of intermittent greening also on the leaf. Enlarged lesions, so these lesions can enlarge, and in the end, the whole leaf can turn brown. Plant resistant varieties treat seed with hot water. Hot water treatment can be done at 53 to 54 degrees centigrade temperature for 10 to 12 minutes. So this can kill the bacteria. Keep your fields clean. Use balanced amount of plant nutrients. So these initial first 4 or 5 points may be common to all kind of diseases. As a student, you can remember these are very fundamental things that use plant resistant variety, treatment of seed in hot water, or treatment of seed with fungicide, keep fields clean, use balanced amount of plant nutrients. So these are generally common points to all management of all the disease. Because in management, you talk about cultural practices, you talk about mechanical methods or physical methods, or some kind of chemical control, biological control, and so on. Wait a moment. So management here means integrated management of disease, ensure good drainage of fields, drain the field during severe flood. So there should not be excess water in this case. Dry the field during the fallow period to kill the bacteria in the soil and in plant residues. So drying of field during the fallow period. However, in very few areas you get fallow period. Of course, we have got rice fallows. Rice fallows, how much area is there under rice fallow, and which part of the country they are found? Rice fallow. Anybody have any idea? What are rice fallows? What do you mean by rice fallow? And in which, how much is area under rice fallows? And in what part of the, which part of the country you find rice fallows? Yeah, anybody?
Sir, in Eastern India.
Yes, you are right. But you can add South India also. It is not just Eastern India. It is in South India, Eastern India, and also in Central India. Rice fallows. So you can remember it is in Eastern India, some areas in Central India and South India. So rice fallows are the land where you get continuously this flooding of water. And after harvest of rice, there is no drainage of water from those soils. So it gets very difficult to do sowing of the Rabi crop or next crop. So therefore, the most of the area remains fallow. So those kind of land where only rice is grown, and after harvest of rice, it is mostly wet soils. Therefore, sowing of next crop is not possible easily. So those kind of soils are called as rice fallows. And the area under rice fallows may be 10 to 12 million hectare in the country. And many times people speak about growing oil seeds and pulses in rice fallows. So people are targeting those, and people are developing varieties of the crops that can suit to those conditions. So that, for example, Utera cultivation can be practiced. You have wet soils and you have no time to plow it, no time to sow the seed in lines, no time or no machinery to do the sowing under wet conditions. So one can broadcast the seed, and Utera cultivation is possible, but only under 100% saturated soils. But if you have standing water, these oil seeds and pulses cannot be grown on those soils. So you should know little bit about rice fallows. So this ensure good drainage of field, drain the field during severe flood, dry the field during the fallow period to kill the bacteria in the soil and in plant residues. Use copper-based fungicides at heading stage. So in the last case also, you have seen agromycin 175 gram and 500 gram Blitox, copper oxychloride. So here also you can use the same medicine or same chemical to treat this disease. Now, paddy blast. Paddy blast is another important disease, which is very common in North India, this paddy blast. And it comes in two stages. One is it comes on leaf, and other is it come on nodes, N-O-D-E-S, nodes. And these nodes can be any node of the plant, of the shoot, of the stem, and it can come on the topmost node, and topmost node is neck, N-E-C-K, neck. You know, the last node where your panicle is attached, or base of the panicle. Base of the panicle is neck, N-E-C-K. So it is called as leaf blast, node blast, N-O-D, node blast, and also neck blast. Neck blast and node blast are almost similar, only with the difference that neck blast occurs on the topmost node, and the other, if it occurs on other nodes, that is node blast. So what are the conditions where this disease occur? Low soil moisture, frequent and prolonged periods of rain shower, means there is no sunshine, frequent and prolonged period of rain shower, and cool temperature during the day. Large day-night temperature fluctuations or differences that cause dew formation on leaves. Cooler temperature favor the development of paddy blast. Disease can occur at all growth stages. It can occur in nursery, and it can occur in vegetative stage, or it can occur in reproductive or even grain filling stages. So it can occur at any stages. In vegetative phase or during reproductive phase, it can occur on leaves, and at grain ripening or grain filling period, it can occur on your nodes, or it can occur on panicle also, which is your base of the panicle. Symptoms, very interesting symptom. You can see on the right side, it is green leaf, and you can see the symptoms. Very interesting symptom and very easy to remember. These spots are a few millimeter in length, and you can see smaller in width, and you can see inside the spot, the color is gray color or whitish kind of color or silver color. And outside on the margin of the spot, you can see color is brown, dark brown, or somewhat reddish brown. So this is symptom, and the size of the spot is just like our eyes. or just like boat, B-O-A-T, boat size or eye size, shape, sorry, this is not shape, shape, eye shape spots or boat size, boat shape spots. So white to gray lesions spots with dark green borders. In the beginning, lesions are elliptical or spindle shaped or eye shaped, and whitish to gray centers. You can see whitish to gray centers with red to brownish or necrotic borders. So these are typical symptoms of paddy blast or leaf blast. Lesions can enlarge later on if the infection continues and the conditions for prevalence of disease are there, then it will advance, go to advanced stage, and the size of the spots will enlarge and they will coalesce. Coalesce means they will merge into each other and growing together to kill the entire leaves. So in the end, entire leaves can turn brown and there will be death of the leaves. So these are the symptoms on the leaf. Now you can see symptom on the leaves, old lesions. On the right picture, you can see old lesions. Once the lesions become older, then you cannot have two different colors. They all becomes brown. And you can also see some grains here. So grains also have some brown color. So quality and quantity both of the grains are affected here. On the left side, you can see the picture. This is your collar or neck blast. Here, the neck region of the shoot or of the stem is affected. So you can see rotting occurs. And what happens? This particular panicle will bend. It will bend down because this neck will become weaker. And within a night or within 24 hours of infection, all the panicles can bend down. They can bend down, and you will surprise that within few days your crop can be devastated. I've seen certain fields of the farmers where within 24 hours their crop was in loss. Most of the panicles were upside down. So sometimes it can be very severe disease. And these symptoms are just diamond-shaped lesions on the leaves, you can see. Now, neck blast or node blast. Lesions on nodes, blackish to grayish brown. Nodes can cause gum to break. It can cause gum to break. Gum can break due to nodes. It can break from the central nodes or from the upper node. Lesions on neck are grayish brown and can cause girdling. And panicle fall over means panicle will hang. Panicle will hang, H-A-N-G, hang. If infection before milky stage, no grain formation. This is very severe neck blast, or it can cause white heads. So sometimes there may not be falling or there may not be bending of the panicles, but they will turn white because there is no grain filling. If grain filling is not there, the grain will turn white. Spikelet will turn white, and similar to stem borer infestation. Stem borer infestation, I may touch later, see maybe next day or tomorrow, but cannot be pulled as in ease of stem borer. So in case of infestation of stem borer also, you get similar symptoms. Your color of the ear or panicle is white in both the cases, but in case of neck blast, if you want to take out the panicle by your hands or fingers, it will not come out in case of neck blast. However, if it is affected by stem borer, then those panicles can be easily pulled. You can easily pull out them from the plant. That is the difference. So white heads also appear sometimes due to neck blast. Now you can see here in this picture, this is neck blast. In the neck region, you can see the rotting, and then panicles are bending down. Panicle is bending down. See, it bends like this. In neck blast, you can see the panicle is bending like this. And on the right side, you can see the grain size and their discoloration of grains. Now this is node blast on the nodes, NODES, nodes. Now management, as usual, resistant varieties, balanced fertilization, but here this silicon application is important. Silicon fertilizers, I just told you some other day that silicon is very, very important, and this reduces the incidence of paddy blast, the silicon. So silicon can be applied in soils wherever you have deficiency or wherever silicon is not available to the crop. And you already seen silica percent in rice straw. Seed treatment can be done at 2 gram per kg seed with captan or carbendazim or thiram or tricyclazole. So any of these fungicide can be taken, but carbendazim is normally preferred, which is also known as bavastine. And spraying of tricyclazole can be done at 1 gram per liter or adifenophos at 1 ml per liter or carbendazim at 1 gram per liter. So tricyclazole can be used at 1 gram per liter, and it is very effective control. But if you want to export your rice, if it is basmati rice or any superior quality rice, and you want to export to other countries, then there should not be any residues of tricyclazole. Many countries do not like that tricyclazole reaches their rice crop. So for European Union, the maximum permissible limit of tricyclazole is 0.1 ppm. 0.1 ppm. If your paddy consignment contains more than 0.1 ppm of tricyclazole, it will be rejected and sent back to India. For Japanese, they can tolerate up to 3 ppm of tricyclazole. So this fungicide should be used carefully. And after flowering stage, it should not be used. If it is used after flowering stage, or you can say heading stage, then it can be detected. But if tricyclazole is used during vegetative phase, then it does not leave residues. So farmers, if they are interested, they can use it, but before heading stage. Better to go for carbendazim or bavastine, 0.1%, that means 1 gram per liter. Now, brown spot is very much important, and it has been historical disease. Major factor contributing to the Great Bengal famine in 1943. This brown spot had devastated rice crop in West Bengal and some other parts of the country. So it infects the coleoptile, leaves, leaf sheath, panicle, branches, glooms, and spikelets. So it can be seen on any part of the plant, starting from leaf, stem, sheath, panicle, branches, glooms, and all the parts of rice, I think except root, can be affected by brown spot. So when infection occurs in the seed, unfilled grains or spotted or discolored seeds are formed in spikelets. Fungus can survive in seed for more than 4 years. So fundamentally, it is a seed-borne disease and is spread from plant to plant also through air, through spores. What is the causal organism of brown spot?
Helminthosporium.
Helminthosporium oryzae.
And for other, this paddy blast?
Pyracularia griseum.
Pyracularia oryzae.
Very good, very good, good. You are attentive, that means. Now, symptoms. Most critical during empty means maximum tilling to ripening stage. You can see on the right side, they are oval or rounded spots. Fully developed lesions means spots. Lesions are circular to oval with a light brown to gray center, surrounded by a reddish brown margin. Infected glooms, panicle branches have dark brown to black oval spots. Infection of florets leads to incomplete or disrupted grain filling, spotting, and discoloration of grains. So these are the important symptoms of brown spot. Balanced nutrition and soil low in silicon. Apply calcium silicate as a source of silicon. Use resistant varieties. Treat the seed with hot water. So you can see this hot water treatment is good for many bacterial diseases also, and for this kind of fungus also. So 53 to 54 degrees centigrade, you can remember, for 10 to 12 minutes. Seed treatment with Captan, Therum, or even carbendazim can be helpful. Spray Mancozeb 2 gram per liter or Adifenofos 1 ml per liter 2 to 3 times at 10 to 15 days interval. False smut, very interesting disease, and it does not come every year. It comes sometimes when it gets favorable conditions. Certain favorable conditions may be there for this disease, and it does not come in on all the plants. It comes in means sporadically, means here and there in the field, but and it cannot be very serious disease, but sometimes it reduces the quality of the grain. Chalkiness of grains can occur, reduction in grain weight, reduces seed germination. If you use the seed infected by such fungus, and then your seed may not germinate. And what are the predisposing factor? Rain, high humidity percentage, and soils with high nitrogen favor this disease. Wind can spread fungal spores. You can see on the right side, fungal spores visible only after panicle exertion, can infect the plant during flowering stage. So this is very interesting disease, that the symptoms of this disease will not come on leaves or any other part of the plant. The symptom will come only on grain. When your heading occurs and grain development start, during that period, you can see the symptoms. However, fungus is residing, fungus is living in the whole plant system, but it is not showing any symptom. It will show symptom when your heading stage occurs. So that is why its identification is not easy. So, and also interesting fact is that the grains, you can see the size of the grain will become 2.5 times more than the normal grains. And the grains are full of fungus balls or spores. And initially the color is, you can see symptoms on the next. And I want to ask you why it is called as false smut. Why it is called false smut?
Sir, it is actually look like एकदम
Not
स्मट की तरह ही रहता है, लेकिन जो कॉज ऑरिजिन रहता है सर उस फैमिली से बिलोंग नहीं करता है ये.
Exactly. So which are the smut genus of fungi?
Ustilago usta gynoidiferens.
Very good. You are very well informed people. So Astilago is true smut, and there may be some more species that can cause smut, but it is not true smut. Therefore, false smut. Good. Grain transformed to a mass of spore bars. Spore bars initially orange. You can see on the right side when the infection starts. So during the initial phase, the color is yellow to orange. You can see on the right side. Then after a few days, the color will change to greenish black. You can see the color change. So not all spikelets of a panicle may be affected. And spore bars initially orange turn greenish black. Sorry. Individual rice grain transport into mass of velvety spores or yellow fruiting bodies, and only few grains in panicle are usually infected. For control, keep the field clean, remove infected seeds, panicles, and plant debris after harvest. Use certified seed, treat the seed at 52 degrees centigrade for 10 minutes. Carbendazim is your Bavastine, 2 gram per kg seed, and same Bavastine can be sprayed, but 1 gram per liter, 0.1%, and spraying copper oxychloride. It is preferable over other fungicides. This copper oxychloride, 2.5 gram per liter, or propiconazole at 1 ml per liter at boot leaf and milky stages. If you suspect that fungus will certainly come, then you can do it in advance. So this was about false smut. So we will start this sheath blight after the break, because now this link is going to be over in next 1 minute. So we'll go to next link. Now it is turn of sheath blight. As the name suggests, blighting occurs. Blighting means just kind of burning or drying. And sheath means, you understand, I explained you too much about sheath. It is part of the leaf, which is partly covered on the cum or rice stem. And sheath blight is caused by Rhizoctonia solani. Leaves dry out and die rapidly. Young tillers can also be destroyed. So sometimes it can be very severe disease, particularly when you have a very dense population. Your growth is too much tilling and excess growth. So occurs in areas with high temperature, 28 to 38 degrees centigrade, high levels of N fertilizer, and high levels of relative humidity, 85 to 100%. And also high seeding rate or close plant spacing and dense canopy. So these are the kind of preconditions that will increase the incidence of sheath blight. So on the right side, you can see this picture, oval, greenish, gray lessons, and there are irregular lessons here. Lesions. I don't know how to pronounce lesions or lessons. Lesions. 1 to 3 centimeter in length, you can see, expand to the upper part of the sheaths, the leaves, and then spread to the neighboring tillers. So you are seeing it on the leaf blade part, but in the beginning, it was on the sheath. So from sheath, it can spread to the whole leaf, but primarily it is concentrated to the sheath part. And you can see this picture. This picture I took 2 years before from a PhD student's experiment. So this crop was, you can see the situation. It is totally drying. So initial symptoms came on the leaf sheath, and then leaf sheath dried up. And once the sheath is dried, the whole blade will also dry, because leaf blade is getting its nutrients, water, everything through the leaf sheath. If your leaf sheath is affected, it is drying, then ultimately your leaf blade will also dry, because it will not get food, it will not get water, or it can also get infection. So sometimes leaf sheath sheath blight can be very severe disease. Use reasonable level of fertilizer, means balanced fertilization. Use reasoned density or reasonable density or optimum density of the plant. Control the weeds. Drain rice field relatively early in the cropping season. Use fungicide to treat the seed. And carbendazim or bavestine 1 gram per liter. Propiconazole 1 ml per liter. Now, Tungro virus, as the name suggests, it is caused by combination of 2 viruses transmitted by green leaf hopper. So green leaf hopper is among the 5 different hoppers. So 5 different leaf hoppers affect the rice crop. They are brown, white, bagged, and different names of leaf hoppers. And 1 leaf hopper is green, because color of the leaf hopper is green. It causes leaf discoloration. You can see right side. Stunted growth means plant is stopped in increasing in height, reduced tiller numbers, and sterile or partly filled grains. So it can affect any part of the plant, whether it is leaf, whether it is tiller or panicle or grain numbers. Plants are most vulnerable at tilling stage. Susceptible at tilling stage. So sources of virus, what can be sources of virus? Infected rice plants, means old plants, stubbles of previous crops, new growth from infected stubbles. Sometimes you are getting new growth from the stubbles, or you are taking the ratoon crop. Sometimes this ratoon of rice is also possible. Volunteer rice plants, infected plants in nearby rice fields, and seedlings raised in infected nurseries. So they can be transmitted or transferred from one field to another field by green leafhoppers. Symptoms, you can see more symptoms on the picture here. Yellow or orange discoloration begins from leaf tip and extend down to the blade. Leaves show mottled or striped appearance, rust-colored spot, and interveinal necrosis. Necrosis means death of the tissues. Delayed flowering, maturity, reduced tillers, incompletely exerted panicles, means they are still covered by the flag leaf. Higher percentage of sterile panicles, and sometimes it is also known as red leaf disease. Tungro virus affected field, you can see the symptoms from here. So it is not continuous. In some cases, you will find green plants and then some are affected plants. On the right side, you can see the green leaf hopper. Management using insecticide to control the leaf hoppers can be controlled. They are vectors growing tungro and or leaf hopper resistant varieties. Adjust planting times to when green leaf hopper are not in season or abundant if known. So this is a way. Plow infected stubbles immediately after harvest. So only this leaf hopper can be controlled by some insecticides. I will discuss when we deal with insect pest management in rice, but the leaf hopper can be controlled by carbafuran, 3G, 30 kg per hectare. Now we move on to next disease, Bakani disease. This is also known as foot rot, FOOT, foot rot disease. You can see on the right side picture, one is normal plant and one is affected by Bakani disease. So plants are relatively taller and they are yellow in color. You can see the plant is showing yellowness, and after some time, these yellow leaves will dry. So seed-borne fungal disease, and what is the name of causal organism of Bakani?
Gibberella fujikuroi.
Gibberella fujikuroi.
Very good. Who was the scientist who discovered or found this disease? Causal
एक
organism.
जापानीज साइंटिस्ट था सर.
Okay. Name? Can you remember?
फुजिकोरी ऐसा कुछ था सर.
Yeah, must be some Fujikori. That is why the credit is given to his
Yes sir. That's why his name is Fujikori.
Good. So this Bakani is really, it is endemic in some areas. It continuously occurs year after year, Bakani disease, particularly in some basmati varieties, and also in southern India, eastern India. It comes sporadically, not very regularly, but it is there. This disease can come, in some cases, it can be very severe. We used to have one variety from IERA, basmati variety, Pusa Basmati 1 and Pusa Basmati 1121. They have been very, very popular varieties. But Pusa Basmati 1 became susceptible to Bakani, besides some other diseases that became susceptible to Bakani after 20-25 years of cultivation. So you cannot keep a variety continuously resistant to all the diseases. With time, they can become susceptible to some other diseases also. So same is the story with 1121. It is still being preferred by farmers, they grow it, but they have to face the Bakani incidence in Pusa 1121. But management options are available so that you can reduce the incidence of Bakani disease. So it is seed-borne fungal disease, and fungus infects plant through the roots or crowns. Infected plants means the fungus is distributed into the plant. It grows into the plant. Infected plants are abnormally tall with pale, thin leaves, produce fewer tillers, and produce only partially filled or empty grains. If first thing is that there may not be heading. If heading occurs, then the grains will remain empty or unfilled. It spreads through wind or water also. The fungus can spread through wind or water also. So management options, you can see these pictures on the extreme right side. This was even after transplanting. Just after transplanting, you can see this picture, extreme right. And this is Bakani disease affected plant, which is taller than the rest of the plant. It is not Kamal of photography. It is actually Bakani affected plant. Just one plant you can see. So that means the nursery was infected. Therefore, during early establishment phase, how come this fungus can make your plant taller? So it can begin with the nursery area. It can come to the tilling. It can appear in tilling stage or any stage. It can show symptoms. So what are the management options? And you can see in the central picture, the whole hill has dried up, and some plants are taller than rest of the plants. So use clean seed to minimize the occurrence. Seed treatment with carbendazim, Bavastine can be done. Seed treatment, I have told you how to do it with the Bavastine, and difficult to control in field. There is no any management option to control this disease in the field. What you can do, if you recognize, identify plants in tilling stage or after transplanting, remove those plants carefully and bring them outside the field and put them into a pit and cover them or either burn them. So that is the only way. There is many times people mislead farmers that you spray this and that, but there is no practically control measure if the disease is in the field. If it has come to the main field, there is no chemical to control this disease. Now, stem rot is also important disease of rice. Infection bodies are sclerotia. I think you know, you must have gone through some plant pathology courses. Sclerotia found in upper soil layers. So these are the surviving part of a fungus, sclerotia. And through them, they survive in soil and also survive on straw buried in soil. The sclerotia float on irrigation water and infect newly planted rice. Then they will get revived, or they will get life. This sclerotia will make some spores or something so that infection can occur. So infected stems rots, सड़ते हैं, they start rotting. Visible numerous tiny white and black sclerotia and mycelium inside the infected comes. The easiest identification of this is that you uproot the plant, remove the leaves, and then you how to say it, आप उसको stem को फाड़ते हैं, चीरते हैं. You make two part longitudinally, cut the stem longitudinally into two part, and inside the stem you will find sclerotia through your eyes. You don't need microscope. You'll find sclerotia and mycelium inside the stem, and which is gray in color. Gray color mycelium and black color sclerotia are visible. So visible numerous tiny white and black sclerotia. White is mycelium or gray, and black sclerotia. Infected come lodges and causes unfilled panicles and chalky grains. Initial symptoms are small, irregular black lesions on the outer sheath near water level. So it starts from the lower side of the plant, stem rotting. Lesions expand as the disease advances. Severe infection causes tiller death. Use resistant varieties, remove crop residues. After harvest, let the store decompose, means cleaning of the field. Drain the field to reduce sclerotia. You can drain the water and put the fresh water in the field. Balance the use of fertilizer with high potash. Spray suitable fungicide. Your Bavastine or propiconazole can be used. So you can see these pictures of stem rot on the all the three pictures, and very clearly showing you. So whenever you see black area on the nodes or on the near the sheath, then this is your stem rot. root knot nematode. So it can be under the animal, it can be under your entomology. I think root knot should come in the entomology. Anyway, it is not, it will not cause disease, but it will make life of the plant difficult. So here causal organism of disease, or you can say disorder is your root knot nematode. Can we call the infection of nematode as disease or physiological disorder, or should we consider it as insect? What are your views? In which class it should go? Should it go to plant pathology? Should it go to entomology? Or where should it go? Nematode.
Plant pathology.
Plant pathology. Okay. So, but if I take it in entomology, then what is the harm? I'm not contesting your views. I respect your views. Just for a lighter mood, I'm asking it. So I think they have got a different discipline, nematology. So sometimes you study them under in nematology also. Entomology. They say they are nematodes, they are animals. So they belong to entomology. And people say that they are affecting the plants. The plant pathology people will say that we are doing the pathology, the disease of plant, and it is a disease. Of course, it is caused by nematode, but it is a disease. Therefore, it is part of plant pathology. But nematode does not mind. It will infect your plant. It will create problem. It won't mind if you keep him in entomology or pathology or nematology. It does not. He will never mind. Anyway. So root knot nematode organism is M. graminicola. What is M? Graminicola?
Sir, Melidogany.
Melidogany. Good.
Melidogany, sir.
Yeah. So I wrote in brief this M. Melidogany graminicola penetrates through the root tips, and females develop within the root, and eggs are laid in the cortex. So eggs are laid inside the plant roots, and galls are the balls, 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 swellings. So galls are formed in 72 hours of the infection. So the infection will come from soil. In soil, these nematodes reside or live. And when your plants start growing, making roots, then the nematode will enter the root, and it will lay the egg, and it start its growth and making the galls. You can see some galls or some swelling in the picture here. Roots. The juveniles remain in the maternal gall. Juvenile here means young nematodes, or migrate intercellularly. So they can migrate intercellularly, means within the spaces between cells, they can move from one part to another part, from one root to another root, through the erenchymatous tissues of the cortex to new feeding sites. Why they move? They want food. So they move into new feeding site within the same root. Survival. M. graminicola can survive in soil flooded to a depth of 1 meter for at least 5 months. So 5 months it can survive without oxygen. You can see if it is flooded to 1 month. It cannot rise in flooded conditions. So normally, if your soils are flooded with water, you will never ever have root knot nematode infestation, but quickly invades when infested soils are drained. So these nematodes mainly come under slightly wet conditions or dry conditions. Whenever standing water is not there on the field, these nematode will come. Otherwise, if your soils are flooded with water, these root knot nematode will not come. They come mostly in direct seeded rice, aerobic rice, upland rice, kind of rices where soils are not kept flooded. So it develops best in moisture of 20 to 30% and soil dryness. You can see this characteristics hooked like galls on the roots in this picture. Newly emerged leaves appear distorted. You can see right side and crinkled along the margins. Stunting can occur. The plant growth will be stopped, and infected plants lower and mature early. See the picture on the right side. Picture on the right side, and it was taken long back. And in this picture, you can see the growth is stunted and plants are showing yellow leaves. If we do not uproot the plant, if we do not see the roots, then you will say that this is sulfur deficiency, this is iron deficiency, this is nitrogen deficiency, this is viral infection, this is bacterial disease, this is that, this is that. So there can be many reasons for yellowing of leaves, for stunted growth. You cannot be sure that it is affected by nematodes unless you uproot the plant and study the roots. So many times as a student, whenever you go to the field, if you see yellowing of the plants, at least you must observe the whole plant. You should uproot. You may find termite infection on the roots. You may find fungal infection or other infection on the roots, which can be the cause. So half reasons of the problem of the plants lie in the soil. So you must check the soil also simultaneously. Now management is use of nematicides. This carbafuran is insecticide as well as it is fumigant, and it can act as a nematicide. So carbafuran 3 gram per 30 kg at the rate 30 kg per hectare can be used. If your carbafuran is 10 granule, then it can be used at 10 kg per hectare. In the nurseries, these nematodes can also come in the nursery wherever you don't have flooding conditions. So there also you can broadcast this carbafuran at the rate of 3 gram per meter square. 3 gram per meter square or 30 kg per hectare. Treat the seed with carbafuran if you have doubt. Use resistant varieties. Continuous flooding is the answer. And raising the rice seedling in flooded soils and crop rotations. By crop rotations, we can minimize the infestation. And another treatment is 200 to 250 kg neem cake. Under certain conditions, neem cake can be useful. 200 to 250 kg per hectare. Neem cake for nematode management. And other is that somebody can grow marigolds around the field, and that can also help in management of nematode. So you can also have Ophrah nematode. This is Dietylnel D. angustus, and it is found in many countries. You can see the symptoms. It is affecting the panicles. Panicles remain underdeveloped. So plants severely stunted, withered leaves, leaves brown areas near the node. Heads develop partially. You can see on the right side, head development. There are no good number of spikelets. So head develops partially, twisted and deformed. So actually nematodes enter the system of the plant and they can reach the panicles and they can lead to such kind of symptoms. So developing heads may contain many nematodes. So they are there in the heads in all stages from eggs to adult. All cycles are there, all stages are there in the panicles. So management is same as in case of root node nematode. So this nematode, Ophrah nematode, is observed or is reported from eastern India, particularly from Odisha. It is reported, this Ophrah nematodes. White tip disease. White tip disease is also caused by nematode. So this is the third disease. So one of the student was right. He was saying that it is a disease. So I think now we should bring it from entomology to pathology. So better to keep it in pathology. Thank you. White tip disease, nematode. Third disease, and you can read the name of the nematode. It is very complicated name. I cannot remember. I can just say it is A. besseyi Christi. Symptoms: leaves with white tip of 2.5 to 5 centimeter long. You can see tip part become white and twisted and wrinkled. The tips become twisted and wrinkled. Flag leaf may be twisted near the panicle. The infected plants are generally stunted, lack vigor, and produce small panicles. So high sterility in panicles, distorted glooms your lemma palea, small and distorted kernels. So overall, you can see it is affecting the grains finally. So you can see a close picture of symptoms of white tip nematodes, as the name suggests. So white tip nematodes, more pictures. So it is not very common disease, this white tip nematode. Management is resident variety, pre-soaking overnight and sun drying, 6 hours per day, hot water treatment, 52 to 53 degrees centigrade. And carbafuran, like in another nematodes, you can use carbafuran, apply cartap hydrochloride. So you just forget this cartap hydrochloride. Don't use it, just use carbafuran for nematode or neem cake 200 to 250 kilogram per hectare. Now, besides white tip disease, one more confusing disease is there. With the name white, there one more disease is there. White leaf disease of rice. It is known as rice hoja blanca virus. So people credit a particular virus is responsible for this disease, rice hoja blanca virus. It is not very commonly reported disease. It is not very devastating or very severe disease, but for the namesake, it is a disease. White leaf disease of rice. It happens in some other cereal crops also. In sugarcane also, you will find white leaf disease of sugarcane. Of course, not very common. But as a student, you should know what is white leaf disease of rice caused by virus. Name is Hoja blanca virus. Inflorescence symptoms on inflorescence, discoloration panicle, inflorescence twisting and distortion of the panicle, and leaves become abnormal colors, or you can say white color. The young leaves, when they are formed, they will be fully white, and later on they become yellow or dead. Roots, reduced root system, seeds empty grains, stem stunting, and whole plant dwarfing. So you can see the symptoms here. Panicles of infected tillers may be completely sterile with malformed and discolored florets and grains. So symptoms and causal organism is not exactly known for this disease. Simply people are taking some guess and they are naming it. So cream colored elongated spots appear on immature leaves. Juvenile leaves become chlorotic, showing spotting or mottling of the leaf. Chlorotic areas increase and coalesce first into stripes, infected after the flowering stage, and tillers are stunted and there is no control is available. So some snails may also come. They are not very dangerous, but in certain countries like in China, many snails affect the grain yield or growth of rice crop. You can see these snails have come near to the base of the rice plant and they are doing some damage. So two species, you can read the name of the species, Pomacia canaliculata and P. maculata, cause damage, introduced to Asia from South America, eat young and emerging seedlings. So they can eat the young and emerging seedling or tillers up to 30 days. They cut the rice stem at the base. They can cut the stem. Once the rice plant reaches 30 to 40 days, it will become thick enough to resist the snails. So this is in general early tilling stage or early growth establishment stage. It can come, but it is not a severe problem. It is a very minor pest. So it can spread through irrigation canals, natural water distributaries, pathways, and during floods. And when water is absent, bury themselves in mud and hibernate up to 6 months. So they can come under ideal conditions. When water is applied, snails may emerge. Identification: have muddy brown shell and golden pinkish or orange yellow flesh. So you can identify snails. So this was just to tell you that some snails can affect the rice crop. So name is golden apple snail. Golden apple snail, maybe because of egg mass, looks like this. It is reddish in color. The critical time to manage at land preparation, especially first 10 days after transplanting and during the first 21 days after direct wet seeding. So it has got some management options are available. Encourage natural predators. Biological control is also available. You can go through it. So this ends this presentation. Now we have 5 minutes time, and you are welcome to ask questions on disease management in rice.